I'm Daniel Levine, and this is the Bio Report. Antibody discovery is a long, costly, and complex process. LabGenius is reinventing that process by using its platform technology that combines AI with robotic systems capable of designing, conducting, and learning from its own experiments. Its platform not only has the potential to accelerate the drug discovery process, but can generate high-performing molecules that likely would not have been found through conventional discovery methods. We spoke to James Field, founder and CEO of LabGenius, about the company's use of generative AI to discover new therapeutic antibodies, its initial focus on immunotherapies, and why he thinks the data the company is generating to train its AI system is a significant point of differentiation. James, thanks for joining us.
Thanks so much for having me on the show.
We're going to talk about AI, drug discovery, and how LabGenius is using the technology to do antibody discovery. Let's start with the world that LabGenius is focused on. Why focus on antibodies, say, as opposed to small molecule drugs or other modalities?
It's a really great question. So, as you know, the business sits at this really interesting interface between biology and technology, and the mission of the company is to accelerate the discovery of novel, uniquely powerful antibodies. And I'm a protein engineer by training, so I did that at master's and PhD level. But I think antibodies are a really interesting substrate for machine learning and artificial intelligence for several reasons. I think the first piece here is these are exceptionally programmable molecules, and really this is a function of the advent of everything that we've seen happening in the world of synthetic biology. It's now really easy to read and write DNA. So if you want to cook up a new antibody, it's a very, very simple process. The second piece is that these molecules are highly modular. So that means that we really can start to recombine different antibody units to create molecules with really, really quite complex mechanisms of action. So we're talking now not just about molecules that may bind to a target, but will have really sophisticated properties. And you can start to think about this as programming logic into these molecules, just in the way that logic gates started to really underpin a lot of the breakthroughs in computer science. But we're looking at the same sort of thing happening in the world of antibody engineering now. And the last piece, the last reason, is that by working in the area of antibody engineering, you really get the opportunity to pick from evolution's palette. So you can take all of the amazing molecules and components that have really arisen through the process of evolution and start to recombine those. So that's why antibodies as a format, as a modality, is particularly amenable to machine learning. But the flip side of it is there also has to be a problem that needs to be solved. And what we certainly see is for complex antibody therapeutics that have these quite sophisticated mechanisms of action, these molecules are really hard to optimize across multiple different properties in parallel. And that's a problem that machine learning really has an opportunity to solve.
Would you say AI is enabling us to do something with regards to antibody discovery that couldn't have been done through traditional means, or is this simply allowing it to be done faster and cheaper?
That's a great question. And often over the lunch table here at LabGenius, you can hear the same conversation being played out. The way in which we see any antibody engineering challenge whenever we start a program is somewhere in this theoretical space that we call design space. The molecule with that right blend of properties exists. It's just a question of how do you find it. Now, with conventional methods, the real challenge there is you're often limited to searching a very, very small area of that design space. And the impact of that is it gives you a fairly low probability of finding the molecule with the right blend of properties that you need. So you can run a program and you may never end up finding a molecule with the right properties. The beauty of the approach that we're taking is that by modeling very large areas of design space, we can systematically search through it and let the data drive us through that exploration of the design space. And what you end up finding is often very, very high performing molecules, often with very non-intuitive designs. So I would say that the process and the approach that we're taking and the application of machine learning to antibody engineering gives you molecules that you just wouldn't have found using conventional methods. But maybe more interestingly, it creates or turns a process that historically has been quite stochastic and artisanal into something that now is highly systematic. You want to further refine the properties of the molecule, you just run another cycle of an active learning process. And I think that's a big game changer here, and really part of this wave that we see of the industrialization of the antibody discovery process.
What would you say distinguishes LabGenius from others applying AI to drug discovery, which is becoming rather ubiquitous?
You're right in the sense that the application of machine learning and AI to the world of drug discovery, wow, there has been such an explosion in this space over the last few years. I think LabGenius has a particularly interesting positioning in this arena, in the sense that you see a lot of groups applying machine learning to modeling protein structure. You see a lot of groups using machine learning to model protein-protein interactions. LabGenius is a little bit different. Our focus is on the application of machine learning for modeling disease-relevant functional cell-based assays. And there are a few ingredients here that really enables us to do this. But let me start by saying a little bit why I think that's so important. So I think a challenge of machine learning is often the first thing people think is, where's the data set that I can currently use? And they might go to a publicly available data set for some property or another. But most of the really, really interesting questions in protein engineering really need bespoke data sets generated for them. And the reason for that is because the public data sets simply don't exist. So the infrastructure that we've set up here at LabGenius is one where we've brought together a lot of software engineering and automation and synthetic biology in order to generate an ML grade data stream, as we call it, where we can run functional screens, these disease-relevant cell-based assays at very, very high throughput. And then that becomes the substrate on which we use to train our models and ultimately power our active learning process. So you can think of LabGenius as really, again, taking something that creates a lot of therapeutic value, these disease-relevant functional cell-based assays, combining it with a lot of software engineering, automation, and synthetic biology to bring online this ML-grade data stream, and then using the combination of the two of those to solve these what are currently intractable problems, so very, very hard protein engineering problems. And I think that's what sets LabGenius apart.
How do you determine the targets or indications you're pursuing?
Yeah, that's a great question. I think a challenge for a company like LabGenius is where you've made a big capital outlay to create a platform that not only spans the world of bits, but also spans the worlds of atoms, is that once you've created that ability to generate an ML grade data stream, you have to be really mindful around the application area into which you apply it. So I think a challenge, as I say, for a business like us is you have to be exceptionally focused. And the focus for LabGenius is on these multi-specific, multivalent antibody therapeutics with quite complex mechanisms of action. For our own internal pipeline, we're applying that to the development of a novel immune cell engagers for the treatment of solid tumors. But certainly in the context of some of our partner programs, we've operated in areas outside that. Now, with respect to the target selection strategy, again, that's a challenge that not only kind of takes into account some of the nuances of being a small biotech, and thinking about that in the context of the financing strategy and also the application of the machine learning. So it's bringing those pieces together that then sets the target strategy. So as a protein engineering company, what we're certainly looking to do, and especially this is the case for our first few molecules, is to work out how we manage that biological risk. And the way in which we do that is we look at in the context of these immune cell engagers, very well biologically validated targets, but looking at targets for which the therapeutic potential of these targets is as yet unrealized because of an issue or that can be solved with a protein engineering solution. So just to make that very, very concrete, for our immune cell engager pipeline, we're really looking here at targets that are differentially expressed on healthy and disease cells, but the exploitation of them therapeutically has been limited by on-target off-tumor effects. And then we're really driving the platform to create molecules that are able to distinguish between healthy and diseased cells with exquisite selectivity. And if I just kind of give you an example here, what we found with the platform is we're able to, through our discovery process, identify molecules with killing selectivities that are at least 400 times better than clinical benchmarks. And that's not just optimizing them for killing selectivity, but crucially co-optimizing them for that in conjunction with other key properties like developability and activity. And that's really where I think the machine learning comes into its own, when you're trying to co-optimize these molecules across multiple different properties in parallel.
Do you begin with a screen of a library of antibodies? Do you create your own antibodies to screen? What's the starting point?
Yeah, we divide the antibody discovery process into two distinct phases. So there's the first phase, which is the lead discovery phase. And really the goal of that is to generate really diverse panels of parts. And then the second stage is the lead optimization phase. And in the lead optimization phase, we're really using the active learning process to identify the best multivalent, multi-specific designs through, as I say, an active learning-led exploration of the design space. So if I kind of give you an example of a program and kind of talk you through the individual steps. Most of the time, we'll start off with a target. And then against that target, and typically there we're looking at a tumor-associated antigen that will be heavily upregulated on a disease cell, but also there present on a subset of healthy cells. What we really need there is a diverse panel of binders. And we'll typically generate those through a combination of traditional methods like immunizations, phage displays, and also deploying some newer generative methods as well. That gives us a pool of parts. Now, in contrast to a lot of traditional antibody discovery processes, we're not interested in just finding the tightest binders. For the sorts of molecules that we're interested in making, the reason they're so hard to engineer is it's often the case that actually maybe like a looser binder might end up being the best subpart in an overall design. So what we really need is a diverse set of developable binders. And those are the sort of, you can think of those as the building blocks that then go into a larger molecule. And then the next step is a lead optimization phase. So we look at this combinatorial mix of parts. And just to give you an example here, we can look at design spaces of over 250,000 T-cell engagers and that design space being described by the combinatorial mix of all of the individual parts. And then we use the active learning process to systematically explore that space. Now, just to compare that to what's done currently, if you're at a leading biotech or pharma company, best case scenario, you're probably only testing 500 to 1,000 of these molecules. So what's really exciting is the sorts of approaches that we're taking here enable you to really get at least an order of magnitude improvement in the number of variants that you can evaluate within a specific period of time. And that's what makes this approach so powerful.
And what data are you able to generate? What's the ultimate goal in identifying antibodies? Is it specificity? Is it affinity? Are there other qualities you're seeking in an ideal candidate?
Yeah, that's a really good question. And it really speaks to the heart of what we're doing, which is trying to build a system that's capable of co-optimizing these molecules across multiple different properties in parallel. The traditional method of engineering these molecules is you'd optimize for one property. Let's say you'd optimize it for some measure of functionality, maybe its potency, efficacy, selectivity, etc. And then when you test it again, you found that accidentally you de-optimized for several other properties. And historically, that's what's caused long timelines in these discovery processes and ultimately made for an inefficient setup. The goal here is to build computational models for each of those different properties. And once you have those models built, you're then able to optimize across them in parallel. So practically what that means for the work that we do here at LabGenius is we have lots of different measures of developability for these molecules, and additionally lots of different measures for functionality. And we run all of the high throughput experimentation on the robotic platform here, generate that data, build models for each of the properties. And then once we've built those models, we can optimize across them to find molecules that are high performing across all of those different properties in parallel. And ultimately what that gives us is these very diverse, highly optimized panels of molecules that then can be progressed into the later stages of the discovery process.
At the end of the day, do you understand all of the variables in the data that the system is looking at? Or is it starting to learn and do things on its own that you may not be necessarily aware of?
Yeah, this is again a really fascinating aspect of the work and the process. So it's often the case that certainly for our early programs, we restricted the design space that we were searching based on a number of biological hypotheses. So in the case of the work that we're doing in T-cell engagers, we made certain assumptions around how wide the immune synapse should be, and therefore how that should translate to the size of the molecule, etc. Certain constraints around how far apart we should space the individual binding domains and what sort of ranges of affinities we should use as well. And then the results we saw were quite interesting in the sense that when we applied all of those biases, we could run cycles of this process and find very poor molecules. And it was only when we relaxed those biases and said, you know what, we're not going to assume anything. We're going to look, trying to reduce as many human bias as possible in this process. What then we actually started seeing is the emergence of very interesting molecules coming out of the process with very interesting topologies that you'd never have designed rationally. Strange combinations of linker lengths and affinities. And really what this kind of hammers home is the fact that you have all of these different levers that you can pull, from topology to affinity to epitope to linker length to valency. And the overall function of the molecule is really this complex interplay of all of those different levers. And the human brain just simply isn't wired to kind of pick that apart. And that's why I think protein engineering as a discipline is so amenable to an approach like machine learning, where actually the truest representation of those relationships is best represented mathematically rather than linguistically. So, of course, when we see an interesting design, you can't help but look at it and try and post-rationalize, I think, this molecule is working in the following way. But I think the reality is these are stories that we tell ourselves. And I think that the truth is a lot more nuanced than that.
That's so interesting to hear you say that. At some point, do you know if you're collecting the right data or if you're collecting extraneous data that may not help identify the best molecules to pursue?
There are two interesting aspects to that. I guess I'll talk first about collecting the right data. A lot of the time to feed these models, we do need a fair amount of data. So at least a couple of hundred designs characterized per cycle that we do. And a challenge with that is it means that the assay is often a proxy of the actual property that you're really interested in. So when we're talking about developability, of course, what we're really interested in is when you really scale up this molecule and you're producing at industrial levels, what kind of yields are you going to see? What are the kind of properties that you'll see associated with that molecule? Now, obviously, it's impractical to do that. So we're taking a proxy assay and seeing how it performs at a much lower scale. So whether we're creating the right data is really a function of for each of those assays, how representative they are of the actual underlying biological function. But this is where we put a big effort in focusing the platform towards running these disease-relevant cell-based assays. And certainly we've seen that an area that I think you can really go wrong here is running maybe some of the easier, higher throughput assays like binding assays that might give you some interesting results and generate a lot of data, but it may not be that biologically relevant. So that's I think the first piece. So we put a strong emphasis on ensuring that our assays are biologically relevant. The second piece is how do you know how much is enough data? And typically what we do there is at the start of any program, we will demark an area of design space. And just by way of example, let's say we're developing a new T-cell engager. We say, we want to search in the space of 250,000 designs. We could never experimentally test all of those, but we'll test a subset, build a model, and that model should be predictive of that total area of design space. Now, the beauty of that approach is every time you feed the model more data, you can reduce the uncertainty of its predictions. So the beauty of this approach, as I say, is the model will tell you when it's had enough data or whether it still has areas of high uncertainty. So using that process, we were able to understand when we fed the model enough data.
You're a synthetic biologist by training, so it was interesting to hear you at the top of this discussion talk in ways I would expect a synthetic biologist to speak. But to what extent are you able to build intelligence into these molecules to make them act or turn them on or turn them off by certain environments or external stimulus, and really turn them into programmable therapies?
Yeah, I'm glad you touched on that because when I look ahead to the future of antibody engineering, really this is the thing that always jumps into my mind. We're just starting to hear now antibody engineers talk about this challenge of building logic into the molecules. And of course, proteins are the nanoscale actuators of life. They perform complex functions. And really, I think we're just scratching the surface in terms of what we can do with this type of molecule. So my expectation is over the next few years, we'll see more and more groups think about engineering antibodies through the lens of building in these complex molecules that can conduct their own logic-based functions. And just by way of example, and just to kind of underline how powerful these functions could be, you could think of an antibody to just conduct one function. And if it was able to accurately distinguish between healthy and disease cells based on differential cell surface profiles, you can start to imagine the applications associated with that. So if this is a disease cell, then do X, and that could be deliver a payload, pull in an immune cell to kill it, etc. So I think now is the time in which we're seeing this step change in the approach towards antibody engineering, as there's this confluence of different disciplines together. So we're having synthetic biologists look at the problem in conjunction with computer scientists, automation engineers, software engineers, etc. And I think it's at that confluence that we'll start to see some of the next major steps being taken forward in the field.
I think a lot of the industry interest in AI is about accelerating drug development, cutting the cost. I think that would be reductive in the case of what you're doing with it, but have you been able to speed the process of preclinical development at all? Have you gotten to molecules faster than you otherwise would have? Is there cost savings from applying the technology in your case?
There certainly are. But I think probably the biggest lever that we have to create value here is to deliver molecules that are more highly optimized and therefore have a lower probability of failing in the downstream translation process. Because obviously the biggest challenge for us as practitioners within this industry is when these molecules fail at a late stage, that's when the biggest financial impact occurs. So certainly by having a more efficient process, you can deliver the same molecule faster. But the thing that excites me the most is delivering more highly optimized molecules that in theory should have a higher probability of reaching patients.
And how much of that optimization happens in silico, and how much of it becomes a more conventional drug development process?
So certainly for the molecule optimization, we almost think of the work going on in the wet lab as an extension of a computational process. And we kind of think of this through the lens of empirical computation, where you have hypothesis generation being generated in silico, and then that hypothesis testing happening in the real world, and then the data obviously forming the underlying models. And it's through that tight integration of the wet lab and the dry lab that you can run that closed loop optimization process and ultimately deliver these highly optimized molecules. Once you've got your panel, then that slots into the standard set of downstream activities associated with progressing these molecules towards the clinic.
My understanding is the system might also suggest to you the next experiment to do. Does it ever suggest surprising experiments?
Absolutely, and I guess just to walk you through an example, um, the obviously the traditional way in which these antibodies are designed is that at every stage of the process, a human will pore over the results and maybe suggest tweaks to the molecule that may or may not improve it. Our process is entirely algorithmically driven. So the first stage in that process, protein engineers will sit down with our data scientists, and as I say, they'll demark an area of design space that they want to explore. Once that's been done, the whole process just runs itself. So in the first cycle, typically what you're doing is you're trying to uniformly sample that space. And then in the subsequent cycles, you're balancing the number of shots on goal that you have between exploring new areas of sequence space and exploiting certain design patterns that seem to be yielding good results. So this is a very different way of approaching the protein engineering challenge. And as I say, it's after you set the initial design space, it's entirely algorithmically generated in terms of the subsequent sets of designs. And what that means practically is the designs that come out at the end of the process end up being molecules that you would never have designed as a human.
And what's the business model? Is it to carry these molecules all the way through development? Are you partnering? Is it a combination of the two?
Yeah, it's a combination of the two. And like many early stage biotechs, we run this hybrid business model where we're investing behind a pipeline of our own molecules whilst we're simultaneously seeking out partnerships that can expand the scope of the platform. And it's really through the latter set of activities that we hope to drive the expansion of the platform. So just by way of example, when we speak to partners, they'll look at our process and look at our results and say, this is fascinating. Can you apply it to our specific modality of antibody therapeutic? And then it will be through those partnerships that we drive the expansion of the platform.
Have you identified a lead program at this point?
Yeah, so we are progressing one of our own lead programs, and again here the big challenge for us as an early stage company has been selecting a target that obviously ourselves and the clinicians with whom we work have a lot of conviction over, but ensuring that we're not taking on too much biological risk, because ultimately LabGenius is a platform that's defined by the power of a company that's defined by the power of the platform, and a mode of failure that obviously everyone in the business is aware of is you can imagine that the platform performs perfectly well, but it's the underlying biology around an unproven target that ends up resulting in the failure of that molecule. So we really, for that lead program, looking for a target that's exceptionally well biologically validated, but there's a huge protein engineering problem that has to be solved around on target off tumor tox.
So are you focusing on solid tumors as the area for LabGenius's own pipeline?
That's correct. Yeah, that's the focus for us. There's a massive unmet need there. And again, the issue of on target off tumor tox is a particular issue for solid tumors.
So this has been a big challenge for immunotherapies, particularly. Any reason to believe you'll be successful where others haven't been?
It's obviously a really tricky area, but obviously a really valuable one as well. And that feeds into this challenge around target selection. So we don't want to be in a situation where there's some broader problem unrelated to target choice that limits the potential of the molecule. So really going for at least for that first program, a very well validated target where there is a molecule that seems to work quite well, but it's plagued by this issue of on-target off-tumor tox, and that's the problem that clinicians really need solved. So yes, we're playing in the solid tumor space, which comes with its challenges, but we're trying to de-risk that as much as possible.
So one of the challenges for a lot of early stage biotechs that have a powerful platform and is both partnering and developing its own pipeline is both allocating resources appropriately and getting value out of the partnerships rather than just using up the funds they provide to do the work. How are you balancing those different demands?
I think you summed it up really nicely there, where a mode of failure here is you take on partnerships and they don't really provide any kind of long-term value for the business. And that's where, when we look at the different partnership opportunities that we have on the table, they've really got to be driving the expansion of the platform. And so the thing that gets me most excited about a partnership opportunity is where it could provide us the ability to go after a target that we would never have gone after. It provides us the option to expand into, say, a new modality, or alternatively, maybe the partnership brings us a lot of data that we can keep and retain, and then use that data to further refine our models and increase the probability of success for our own internal programs.
At this point, I say most big pharma certainly, and a lot of smaller ones have waded into the area of AI in some respect. I still find, talking to some old school drug developers, that there's some skepticism about the value of AI and the hype versus the reality. Do you see these attitudes changing at all over the years in your discussions with potential partners?
I think that they will change, but at the same time, I think a lot of the folks who are skeptics are quite justified in being skeptical. I think because there's been so much interest in machine learning and artificial intelligence, we've seen the blanket application of these technologies to every single stage and every single problem within the drug discovery process. And if I'm being very honest with you, very candid, a lot of the problems in the drug discovery process, the existing solutions are fit for purpose. And AI or machine learning, it's just a tool, and it's not always the best tool. So my expectation is, this first wave of practitioners applying these methods will quickly find out which are the problems that really need to be solved and which are the ones where the existing solutions are adequate. And then once the really hard to solve, high value problems start to be solved with these methods, then I think the interest and the optimism behind the technology will be warranted. But some of the skepticism indeed that you see within the industry, I think, is entirely warranted.
And how is LabGenius financed to date, and how far will existing financing take you?
Yeah, so we've been really mindful when it comes to building an investor base for the business. When I started out with this company, what I was really mindful of is we wanted to build a platform that would be an enduring, beating heart of the business for years to come, and that takes a certain breed of investor. So we've managed to find and coalesce a group of investors who both bridge the world of technology, but also have significant exposure into the world of biology as well. But not only are they able to kind of bridge these different domains, but they also have very deep pockets as well. So we've been well financed by that investor base to date, which sort of spans mostly Europe and the US, and certainly we have their backing in the financing rounds to come as well.
James Field, founder and CEO of LabGenius. James, thanks so much for your time today.
Thanks so much for having me on the show.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.